Okay, I'm going to talk to you about the functional unit of the myofibril or the sarcomere. Yesterday we talked about that is a small structure in the organ that does a physiological function of that organ. I'm just going to review the sections of the sarcomere. When you look at the sarcomere, you see the repeating pattern, and that pattern is going to create striations that you see under the microscope. Remember, this is specifically skeletal muscle, not any other type of muscle, such as cardiac or smooth. So if I zoom into this one sarcomere, I'm going to see two different types of myofilaments. Myofilaments are just muscle filaments. I see the myosin and I see the actin. Myosin is going to be red in all my drawings, and the actin is going to be blue. Those are the two major myofilaments. All the other labeling is mostly just sections of those types of filaments. So the I band is a section here to here. All of this that I have in green, and that band is mostly actin. And since actin is also can be called the thin filament, that is going to look lighter under the microscope. The area that I have shaded pink in the middle, that's going to be the A band. It's the darker, wider band, and that is going to also be seen under the microscope. It's darker because it has the myosin, and the myosin of the two myofilaments is much thicker because it has these little arms, and these little arms are just cross bridges. We'll get to that point, but they're crucial to the contractile motion of these units. The H zone is between the lack of anything, actually. So it's the absence of actin there. If you're going to cause something to contract and relax, you need to have space for that to happen. So the H zone is actually the lack of actin, so that the actin has somewhere to go when your muscle contracts. So when you're contracting your bicep, all of the sarcomeres on the whole length of the myofibril contract, and that shortens the whole muscle all at once, just like an accordion would. And then you have the M line. The M line is just if you drew a line down the center of a sarcomere, that would be like your midline. And then at the ends, you'll have the Z line. Sometimes it's referred to as the Z discs. Z disc, I've seen it either way. But that's just on both ends of every sarcomere. So it's really just the ends of the sarcomere. So all of this is the structure of a single sarcomere. And if I zoom in on the sarcomere to get more microscopic, so let's say I zoom in here, okay, and that takes me to here, down here. So again, we're zooming in further. So you wouldn't be able to see this under a microscope, specifically microscopes that we would have. So I'm kind of going to zoom into one side here. The red and the blue stays the similar. So red is my myosin, blue is going to be my actin, but actin becomes a lot more interesting. So the actin molecules are those blue molecules here. Okay, they look like this. And then on the blue actin molecules, you're going to see a binding site. Okay, that binding site is going to be covered by this tropomyosin, this thread. Okay. So again, the blue, one of these blue beads is an actin molecule. Each of those has a binding site. When the muscle is not contracting, the binding sites are covered by tropomyosin. And then each tropomyosin is controlled by this purple bead, which is called the troponin or troponin. And then on the myosin, you see myosin heads. Okay, so if I zoom in, I see a myosin head on these little arms, and those are going to be crucial to the contraction of this muscle. Because the whole goal of this sarcomere is to contract, and we're going to have to go through the steps of contraction. So as I zoom out, this is the information about a single sarcomere or a unit of muscle.